Hi everyone, this is lecture 17, chromosome mutations. Material here is found in chapter 16, page 463 to 476. So far we've talked about mutations as they happen to the DNA, for example, point mutations, indels, and what happens to proteins like missense, frameshift mutations. These mutations we're talking about today are bigger in scale. They are still DNA mutations, but they affect whole chromosomes. So let's see how that works. Quiz question number 1. Loss of genetic information can result from which type of chromosomal mutation? Question 2. Which type of chromosomal mutation results in no gain or loss of genetic information? Question 3. Chronic myeloid leukemia results from which of the following chromosomal mutations? So we're going to talk about a few different kinds of chromosome mutations, including deletions, duplications, inversions, and translocations. These all cause structural changes to the chromosomes themselves. Like other DNA mutations, chromosome mutations are generally neutral, meaning that they don't often have an effect on the organism's fitness. This is estimated to be about 1 in 625 cases of translocation, which is a swapping of chromosome fragments. And again, the majority of these are harmless for people. How do these happen? Well, like other DNA mutations, they can be spontaneous rearrangements, or induced by chemical or environmental stressors. Spontaneous mutations occur generally because of a process called chromosome recombination. We're going to talk more about crossing over and recombination when we start talking about cell division. But for now, all you need to know is that chromosomes will come together during the process of meiosis, it's called homologous recombination. The maternal and paternal chromosomes will swap genetic information. And sometimes that process doesn't go right. And so you get problems that occur from that. Sometimes, though, environmental stressors like X-rays, other high-energy radiation will cause breaks in the DNA, which causes chromosomal rearrangements. A lot of chemicals can do the same thing. So, what are some of the examples? Well, one of them is called a deletion. Now, we've looked at deletions on the level of base pairs, but this is a deletion of a large segment of a chromosome, many thousands or anywhere from tens to hundreds to thousands to millions of base pairs in length. This chromosome on the left is divided into 8 segments, segment A, B, C, D, E, F, G, H. Each of these segments is a long stretch of chromosome, could be millions of base pairs long. Segment D, in this case, is deleted. And if it does get deleted, then there's a new connection that's made between segment C and segment E, as shown here on the right. That will lose genetic information since the genes that were there before are no longer there. So it's a loss of genetic information. Here's an example. It's called Kreiduschat syndrome. This is from your textbook. This mutation is called 46,5p dash, or deletion. This picture of chromosomes is called a karyotype. We haven't seen karyotypes yet, but this is essentially the collection of chromosomes from a patient. You'll see that they're arranged by their number, with the longest chromosome, chromosome 1, at the beginning, and the shortest chromosome, 22, at the end, and then the sex chromosomes, X and Y. The circled pair here represents where the deletion occurred. It's on the arm of chromosome 5, and you can see this shortened chromosome here on the right-hand side. So one of the 2 chromosomes has a deletion mutation. This karyotype is known as 46 because there's 46 chromosomes, and the short arm, or the P arm of chromosome 5, has a deletion, so it's 5p minus. A bunch of genes are deleted from this mutation, including a copy of a gene called the telomerase gene, but it's not sure, nobody knows if that's the sole source of the syndrome or not. There's lots of different symptoms that go along with this syndrome, so it's a complicated disease. Now, some mutations can actually cause the gain of genetic information. Here's an example. This is called a duplication. So instead of a region being deleted, now you're doubling up the amount of that area of the genome. Here's a chromosome on the left-hand side that has segments A through H again, that went through a duplication where segments B and C together were duplicated. This can happen in a couple of different ways. The one shown on the left is a tandem duplication, where segments B and C are simply duplicated right next to one another. In the middle is a reverse tandem, where B and C are again duplicated but right next to each other but in the reverse orientation. And then finally, a terminal tandem, where segment A and B is duplicated on the arm, on the extreme arm or the end of the chromosome here. So again, duplications are the gain of genetic information. Now, you wouldn't think that that would cause a problem because it's gaining, you're not losing any genes, but occasionally things do go wrong. Here's an example in Drosophila where a segment of their chromosome is duplicated. This is the X chromosome in flies, and the segment is called 16A. The normal case is one copy of the segment. However, in flies who have a mutation called the bar mutation, they have a duplication of this region. This is shown here with the so-called polytene chromosomes. These are salivary gland chromosomes from the Drosophila, and you can see from the banding pattern that there's one area of section 16A, and the duplication, there's two areas of 16A, and it results in a mutant phenotype. How does that happen? Well, it could be some kind of developmental defect. If you have too much of a gene product, you sometimes have defects just as you would with too little of the gene product. What else can happen? Some mutations don't cause the loss or the gain of genetic information, just rearrange it in some way. Here's an example called an inversion. In this example, segments B, C, and D are all together inverted, flipped around upside down as you can see here. So now segment A is connected to segment D, but in the reverse orientation. Remember, DNA is still 5' to 3', it's just reconnected in the opposite direction. Whether the inversion happens around the centromere or includes the centromere determines whether it's called a paracentric or pericentric inversion. A pericentric inversion here includes the area around the centromere. Another type of mutation that causes no gain or loss but just a rearrangement is called a translocation. Translocations generally come in three flavors. On the left-hand side here is a non-reciprocal intrachromosomal translocation, where a segment, B and C, is moved to a new portion, a new spot on the chromosome. So before it was on the long arm, and now it's now also on the new long arm, but it's between segments F and G. So it's just simply moved to a new location on the same chromosome, so intrachromosomal. The other types are called interchromosomal because they're swapping parts of a chromosome with another non-homologous chromosome. Here's an example called non-reciprocal in the middle, because segments B and C are now being removed from the original chromosome and are now being put onto a new chromosome, one that is not homologous. It's not the same as the original. On the right-hand side is a reciprocal translocation, where the sections in yellow and purple have swapped locations. And so now segment M, N, and O are on the larger chromosome, and A and B are on the shorter one. Translocations, inversions, and deletions and duplications can all cause a problem called a break point. The point at which the chromosome breaks and then reattaches may cause a problem if there's a gene in that area. So let's take a look at an example. Here's a really classic example in chronic myeloid leukemia. It's a type of a cancer of white blood cells. We abbreviate this CML. Here's a translocation that occurs in lots of people with CML. It's a translocation, a reciprocal one, between chromosome 9 and chromosome 22. Chromosome 9 has a gene on it called ABL, ABL. This gene is a transcription factor, and it is very active in white blood cells. On chromosome 22 is a gene called BCR. It is a B cell receptor, and it is again very active in B cells. The ABL gene is important for helping cells divide. That's really important if you're a white blood cell. white blood cells need to divide many, many times a day. The B cell receptor is a very highly expressed gene. What happens is there's a translocation of a piece of the chromosome 9 onto the chromosome 22, which juxtaposes the ABL and the BCR together. This is the break point. Part of the ABL gene is connected now to part of the BCR gene. What does that do? Well, it takes a growth regulatory gene, one that promotes growth, and puts it next to a very, very active promoter with the BCR. It makes this gene become way overactive and makes the cells divide way too much, and thus you get cancer. This is called the Philadelphia chromosome because it's a very distinctive looking chromosome. It's very short, and it was found in a hospital in Philadelphia. So that's an example of a translocation. Here's a karyotype of a patient with CML. You can see that chromosome 9 here is a little bit longer than usual. That's where the arrow is pointing here. And chromosome 22 has the shortened version, the Philadelphia chromosome, as shown here. CML has this karyotype called T922, where the T stands for translocation, and it's a reciprocal translocation between chromosomes 9 and 22. All right, that's all for now. I will see you guys in class.